already have an interaction with him yesterday and during the mentorship sessions. So Dr. Martin is a vice chairman and professor of molecular physiology and biophysics at the Baylor College of Medicine. And he also holds the prestigious Vivian L. Smith Chair in Regenerative Medicine and served as a director of the Cardiomyocyte Renewal Laboratory at the Texas Heart Institute. So Dr. Martin is internationally recognized leader in the field of development and regeneration biology. And over his, through his career, he is already authored more than 150 really high impact paper you can name as including Nature, Science, Cell, etc. So his research has fundamentally enhanced our understanding how developmental process influence disease and the regenerations. So when I was a trainee, like many of you here, and then I got fascinated by the transcription factor and the development biology. So I read so much about paper from his lab, and then which is actually was a build of my knowledge. So I'm very personally actually excited to have him here. So this paper, including the early work, is a transcription factor PITX2, which is a left-right asymmetry and also play a role in atrial fibrillation. And then also he uncovered the pivotal function of the BMP signaling pathway in heart development and also the whole embryo development, especially like BMP2 in the atrioventricular cushion development, which is a key player in the valve development in a later stage. So for last over a decade, he's focused more toward the regeneration, heart regeneration, which is a role of the Hippos pathway in a cardiomyocyte proliferation. So Dr. Martin's lab has demonstrated groundbreaking work in a people pathway, how they play a role in the disease, and also as a treatment. So today, Dr. Martin will share his latest finding and his talk titled Hippo Signaling in Heart Regeneration. Please join me welcoming our guest speaker, Dr. Martin.
Yes. Okay, thank you. Thank you for that terrific introduction and for the invitation to come. I have to say that I've really enjoyed my time here. It's a very enthusiastic and a very positive kind of experience. I really enjoyed the poster switch session. That was cool, and then the innovation that we just had was also very exciting. So yeah, thank you for the invitation and let me participate in this great event. So today I'll be talking to you about playing its role in tissue renewal and in particular heart regeneration. I'll start off with a little bit of background, some of our published stuff, and then the last 15 or 20 minutes or so, it'll be a unpublished story profiling human congenital heart disease patients, a little bit of a newer direction for us. And the goal to do that is to identify patient populations that we can treat with some of our therapies that I'll be telling you about. And those are my conflicts right there at the bottom. So the main problem that we really we're interested in addressing is this problem of heart failure, which as people in this room know better than me, is really a huge unmet clinical need. It's the #1 cause of death, certainly in North America. And some of the numbers are shown on this slide. It's not a great slide, but I like this number right here. 300,000 new patients a year are diagnosed with these advanced stages of heart failure. And in the States, we do about 3,000 transplants. So transplant is really the way to directly address the problem of a bad pump, which is basically what heart failure comes down to. Typically, heart failure is ischemic heart failure. It's not only ischemic heart failure, but the majority of heart failure that we see is because of myocardial infarction, bad diets, as well as genetic causes. And these myocardial infarctions cause loss of millions, and in some cases billions of cardiomyocytes, it's been estimated. And basically the cardiomyocytes just don't come back. The cardiomyocytes don't have any renewal capacity or a very limited renewal capacity. How do we know that? Well, there have been some studies looking at this question in humans, which is a difficult thing to do, obviously. Why is it that the heart muscle, the cardiomyocytes, don't renew, and is the big question. And it's been looked at to get sort of a number to quantify it. It's been looked at by a group at the Karolinska Institute, and a number of papers that they published dating back a few years now. What they did was they did something called retrospective carbon-14 dating, and basically what they did was they were looking at people who were born in the years of the atmospheric. Do I have a? Can you see my pointer? There were atmospheric bombs being blown up into the atmosphere, nuclear bombs during those years. Carbon-14 levels were elevated in the atmosphere, gets into the food chain, and then via the food chain gets into our DNA. And so carbon-14 is in our DNA, and it's basically a pulse chase experiment. And so by looking at postmortem samples for people who were alive in those years, and then looking specifically at the cardiomyocyte nuclei and using mass spec technology, it's just a fancy way to look at the isotopes in the cardiomyocyte nuclei. You can look at the ratios of that carbon-14 to the carbon-12, and then you can make some estimates about when the cardiomyocytes were born. And another way to say that is, what's the rate of cardiomyocyte renewal? Typically, what they found was that when you're young, 20, it's about 1% per year, so very, very low. And this is consistent with all the clinical observations. And then as you age, it drops off to even lower numbers, about half that, more or less. So that's also consistent with the observations that heart failure is much worse as we age. And also, you can make calculations that when you're about more or less halfway through your life, about half the cardiomyocytes were born during development, and the other half were generated postnatally. So very low ability to proliferate is really has been the driving paradigm of why people go into heart failure. Generate, and this is very valuable to the field because it makes us think that if we can manipulate mammals more like these non-mammalian vertebrates, in this case the zebrafish, that we can make a mammal heart regenerate. So the zebrafish do regenerate. If you amputate about 20% of the tip of the zebrafish heart, and then you wait a month or 2 months, you can see that the zebrafish heart will regenerate very well. So that's a well-known paradigm. So again, important, and that was done in the early 2000s by Ken Poss, who's a leader in this area. And then now, circa 2011, a paper from Dallas, UT Southwestern, showed that neonatal mice also can regenerate the heart. And they basically did a similar experiment to what was done in the fish, where they amputated the tip of the mouse heart, so in this case a mammal, and then they waited, and if you do that amputation in the P1 through P6 neonatal period, so right after birth, and then you wait 3 weeks or so, you can see that in that case the heart will regenerate. So in a very short window of time, a mammalian heart, the mouse, will regenerate. If you're outside that window of time, P7 and so on and so forth, you get the scarring. You don't get regeneration. So, but then again, this was an important step forward because it showed us that there is some capacity in a mammal, and there's some anecdotal evidence also that humans have capacity to regenerate, and I probably don't have time to go into that, but all these things made us wonder: are there signaling pathways that are inhibitory to cardiomyocyte proliferation? So is there some genetic reason why the mammalian cardiomyocyte doesn't regenerate? If we can figure that out, can we do something useful to do something for people in heart failure, to make some treatments for those people who don't have a lot of options? We started looking at the Hippo pathway, which at that time was primarily studied in flies, and it was known to be an organ-size control pathway. Here's the control fly, and then here's a fly which is deficient for one of the hippo pathway genes. And you can see that the head has a lot of extra folds, and then also the thorax is a lot bigger than it should be. So it's an organ-size control pathway in flies. What is the hippo pathway? If you just look at the right-hand side of this, it's a kinase cascade, as I have on the title here. The core components of the pathway are shown right here. There's a few different kinases. There's serine-threonine kinases. There are some adapter molecules that modulate the activity of those kinases, and then a downstream effector molecule of the kinases. There are two in mammals, but I'm going to be focusing on YAP. So YAP is typically can be phosphorylated by the kinases. When YAP is phosphorylated, it's excluded from the nucleus, and its activity is inhibited when that happens. So phospho-YAP is inhibited, and it's inhibited by the hippokinases. When the hippokinases are low, either for physiologic reasons or because we've manipulated the hippo pathway genetically, which is a lot of the data that I'm going to be talking to you about, YAP can move into the nucleus where it can interact with transcription factors. It's a cofactor. It doesn't bind to the chromatin or the DNA directly. It needs TEAD factors to direct transcription of the YAP target genes. Many of these YAP target genes, there's a lot, but many of them are genes which are important for telling a cell to progress through proliferation, cell cycle, to progress through pretty much all phases of cell cycle. So I think essentially what the hippo pathway does is it's a stop signal. These kinases are activated by signals, physiologic signals coming from outside the cell. High density is one of those sort of classical signals that activate the kinases. And then when those kinases are active, it inhibits YAP activity. So it's basically a stop signal. It's a strong signal to the cell to stop proliferating. And so the simple conceptual model for the way we've been studying this is to take away different components of the stop signal to try to understand what the hippo pathway does in different contexts in the heart, and also, try to understand what YAP is doing in those same type of experiments. So that's really the simple-minded idea. So the first experiment that we did was to take away one of the hippo pathway components, a molecule called Salvador. It's one of the adapters. I'll talk to you a little bit about Salvador. Salvador is one of the adapters right there, SAV, and it's working right here. So it's not a kinase, but it's an adapter. So it's important for the full activity of the stop signal. The first experiment we did a number of years ago now is would take away Salvador in developing cardiomyocytes. So in the developing heart, using a Cree that takes away Salvador in the muscle, the developing cardiomyocytes. And you can see that just like in a fly, in a developing mouse heart, it's an organ-sized control pathway as well. So these hearts are bigger. This is the mutant, and this is the control. It's about 2.5 times larger, but so that function is conserved between flies, all the way from flies to mice. Taking away hippo, there's still quite a bit of it's still patterned correctly. You can tell which is the apex, which is the base, and they're the atria. It doesn't turn into a big tumor or anything like that if you take away the stop signal. This encouraged us then to get back to this question of adult human heart failure, and so we did a simple experiment a number of years ago. This was now, and we just looked at hippo activity in human heart failure, looking at people in end-stage heart failure. They're getting transplants. And we got their tissue, and a simple way to look at hippo activity is to look at phosphorylation status of YAP on a Western blot. The kinases will phosphorylate YAP and inhibit its activity. And you can see that in the patients with heart failure, there's a lot more phospho-YAP. It's about a 3-fold increase. So in a context of a failing heart where you would prefer to have less of the stop signal, you actually get more of the stop signal through reasons and mechanisms that we have been studying now for a while, and we're getting closer, but we still don't totally understand. But anyway, what we did then when we saw this was we decided to do a very large mouse experiment. And this is a paper we published back in 2017, but we infarcted hundreds of mice at that time point 0, and then we waited 3 weeks. And at the 3-week time point, we looked at the mice to make sure that they had gone into heart failure. We didn't do anything genetically, didn't manipulate them in any way. And if they had gone into heart failure, and we measured that a number of different ways, then we, at that point, we inactivated, in some of the mice, we inactivated the hippo pathway, in this case, Salvador, take away the stop signal, or left the stop signal intact, a lot of control experiments at that 3-week time point when they were actually in active heart failure. And then we followed the mice out in a longitudinal study over a period of a total of 9 weeks, and we did a lot of different studies in between there. And we're, just to remind me to tell you that we are taking away the stop signal only in the cardiomyocytes in this experiment. So it's a cardiomyocyte-specific CRE. By ejection fraction, and if you just look at the ejection fraction on the right-hand side, here's time 0. We infarct the mice. We wait 3 weeks. They drop function. We did a lot of other studies at that time point to make, to show that they had signs of heart failure, like pulmonary edema. They gain weight and things like that. And then you can see that the hippo, the group where we've taken away the stop signal, the hippo knockout, Salvador knockout group, over the period of 6 weeks more, they recover their ejection fraction up to that of the unoperated controls, which are these top groups right here. The control that still had the hippo pathway intact doesn't improve function, so they stayed pretty much flat. Because this really tells a lot of, there was a lot of insights, just looking at the histology. So this is a 9-week time point, and you can see the controls have, they don't look very good. There's not a lot of muscle in the free wall of the left ventricle. They're a little bit dilated. You can, if you look at this middle group, this is the knockout, Salvador knockout. So we've taken away the stop signal specifically in the cardiomyocytes. You can see there's a lot more muscle in the free wall of left ventricle, and there's 2 other things. The fibrosis looks quite different. And likewise, there are more endothelial cells that I'm not showing you, but there was an increased microvasculature that came along with these new, what I'm going to call new cardiomyocytes. And so really, it's not just more cardiomyocytes, but we are changing the whole microenvironment. It's a true regenerative microenvironment in which these cardiomyocytes, the way we think about it is these cardiomyocytes that have lost hippo now are actually signaling to the other cells in the microenvironment to promote regeneration of the tissue, the whole tissue. And that's what some of the things that we've been following up more recently. Just to quickly get to this idea that we can use some of the things that we're learning in the lab and to try to develop therapies for people with heart failure. We developed a gene therapy using RNA interference to knock down the hippo pathway, knock down Salvador in this case. And the basic idea, again, is very simple: to reduce the hippo pathway, to reduce the stop signal. And we're using an AAV9 capsid. AAV is adeno-associated virus capsid. And it's driven by a cardiac-specific promoter. And then we have 3 shRNAs against Salvador encoded by the transgene, and it's all in the capsid, AAV9 capsid. Our strategy for humans is to use a catheter-mediated delivery. So thread a catheter up into the heart, and using electromechanical mapping. In some of our earlier experiments, we used NOGA mapping to figure out where the border zone is, and then deliver small doses of this virus into the border zone cardiomyocytes, right around the ischemic area, around the infarct. And so that's really our path forward with this treatment, this gene therapy at the moment. What we think happens, and this is based on our data as well as data from other labs, is that when you inactivate the hippo pathway, that you can induce a de-differentiation. Really, the way we think about this now is more of a sarcomere disassembly. So as you know, the cardiomyocytes have the sarcomeric structures, which allows them to pump. And there are very specific, we think of it as a very specific genetic mechanism to cause those sarcomeres to disassemble to some extent. That allows the cardiomyocytes to re-enter the cell cycle. In addition to having YAP in the nucleus, they've got sarcomeres disassembled, and then there is a cell division. There's a component of cardiomyocyte division going through cytokinesis, and one cardiomyocyte dividing into 2. and then redifferentiation, which is less well understood. The cell division thing, it is depending upon what kind of a manipulation of the Hippo pathway, we can see either a lot of cell division or just smaller amounts of cell division. And for this Salvador knockdown, it's a relatively mild knockdown of the pathway, so we only want to see a relatively small amount of cell division. I'll get to that, I think. I have that slide. Anyway, so doing is we thread the catheter up through the femoral artery, and it's a subendocardial approach, and then the mapping with the NOGA allows us to clearly see where we're doing those injections. And so that is it's a very nice approach, and it allows us to use very low doses, order of magnitude less virus than you would have to use if you were using systemic delivery, and that's important for safety as well as manufacturing challenges of making a lot of virus. So this is the pig study. Basically, it's conceptually very similar to the mouse study, except now we're using pigs. This is the path to the clinic. So we infarcted pigs at time 0, and this is an ischemia reperfusion of a pig, just like what happened to a human. Ischemia reperfusion time 0. We wait 2 weeks for the pigs to go into a reduced pump function. We couldn't actually show that the pigs were actually in outright heart failure, but they definitely had a lot, reduced ejection fractions at that 2-week time point. And then we followed the pigs out for a total of 3 months. And just like in the mice, we do a lot of different studies in between to try to understand what's going on. And really, if you look at the functional studies down below, B, C, D, and E, if you look at E, this is where we're presenting the data as a change in ejection fraction. Here's time 0, where we've performed the MI, the ischemia reperfusion injury in the pig. We wait 2 weeks, and at the 2-week time point, if it's been a good adequate MI, then we go in and deliver the virus using the mapping and the catheter. The control pigs, as you can see, continue to remodel and lose function over the period of the experiment. But in contrast, where we've taken away the stops, reduced the Salvador activity, so reduced the stop signal, there are two different doses of virus. This is the low dose, and this is the high dose. And so they start to improve function relatively quickly, and they start to climb that curve. And so we followed them out, that basically climbed the hill, and followed them out for the 3 months. And so this was a very happy experiment for us to see that, especially after waiting 2 weeks. We've done a lot of studies, but I just will tell you, we do see reduced fibrosis, so remodeling is improved in these pig hearts. Here's control, and here's a Salvador, and these are slices through the control. You can see this pig had a nice anterior infarct, and this is a representative view of one of the slices through the treated pigs where we've knocked down the Hippo pathway. I should also say this is double-blinded. So these studies are done by the Texas Heart Institute Large Animal Facility, by the people who work in that facility, and they are not aware of what the Hippo pathway really is, and they're not connected to these studies in any way. So that's also, it's important to do these studies in a double-blinded fashion, obviously. So we think that this is really an important pathway to target. So I know that heart failure patients are going to be complicated, and much more complicated than these pigs. And so we're very interested in dissecting this pathway further and to look at this in greater depth and more detail. And to do that, we've turned back to the mice, and we have used one of my favorite models, and that is this transgenic mouse that we have made in the lab, a number of years ago now, called it's YAP5SA. And so I'll just tell you briefly what YAP5SA means. YAP5SA, typically, as I mentioned, the kinases will phosphorylate YAP, and then when they do that, YAP will be degraded, typically, and YAP activity is inhibited by the kinases. What we have here is YAP5SA. All of the serines have been mutated to an alanine, so the substrates sites are no longer there. So YAP5SA, as I say here, is impervious to the negative regulation of the Hippo pathway. So we can activate it in adult cardiomyocytes using this transgene. It's a lock-stop-locks cassette, and we can activate YAP5SA in the adult mouse cardiomyocyte acutely just by delivering tamoxifen, and then see what it does in that context to the cardiomyocyte. It's a very strong manipulation of the Hippo pathway. It's sort of like our sledgehammer in the lab. If we really need to see something, this is what we turn to. We know some things about how YAP5SA works from our 2019 study. So we know that YAP5SA can go into the nucleus and bind to the YAP target sites, and in many cases, it actually opens the chromatin. And how do we know that? It increases chromatin accessibility. So here's some these are genomic tracks for an experiment called Assay for Transposase Accessible Chromatin. It's just looking at open versus closed chromatin, and the peaks on these tracks are areas of open chromatin. And the control, as you can see on this gene, noggin, that there are a few open places in the genome, just in the control situation around noggin. In the YAP5SA cardiomyocytes, so these are the cardiomyocytes specific, we see that there are a lot of new places that are opening up. And those new places are strongly enriched for TEAD elements. So they're places that YAP would normally be binding. It's probably at some point, probably during development, is what our data would say. Other genes, likewise, LATS2, which is actually one of the Hippo pathway kinases, you can see that these new places are opening up when you express YAP5SA. And then this is just a computational method to look at around the TEAD elements, and you can see that there's a nucleosome-free region in YAP5SA that's not there in the control. So YAP is actually it's a very powerful molecule. It can go in and open up very specific places in the genome, and it induces a more fetal-like cell state in the cardiomyocytes. And so we had known that from our 2019 paper, but we wanted to get back to this question of the microenvironment. As I mentioned to you, we saw that there were more fibroblasts were different, the endothelial cells were different. So we used this methodology, which is not new anymore, but at the time, I guess there was a point when it was new. But now everybody is using spatial transcriptomics. It's an ideal way to look at the microenvironment, to what's happening in the whole tissue at the same time in a unbiased and genome-wide fashion. So you have your tissue on a section, and we're using the Visium platform. So the Visium platform is actually spots. So it's not true single-cell resolution. So we do single nuclear RNA-seq or single-cell RNA-seq at the same time, and that allows us to deconvolute each spot, and we can make estimates about what are the cell types and the cell states that are in each spot. And it actually works very well. So if you have good single nuclear RNA-seq data to combine with your spatial data, then you can make very good estimates and very accurate estimates. What does it tell us? Well, one of the things, we get a lot of very nice predictions, strong predictions about cell-cell communication and interactions. And you can make models for how macrophages are interacting with cardiomyocytes and pretty much every cell in the heart. You can make those inferences and predictions. You can also see different spatial gene expression across genotypes. So here's a control, and this is a YAP5SA heart. You can see that there's differences between those two genotypes, and then across a region in any tissue, and in this case, the heart, you can see that there are differences. In this case, this gene is highly expressed in the atria. This one is highly expressed in the ventricle. So these are kind of obvious differences, but very, very nice and powerful experiment. One of the first things that we found was that there were indeed two cardiomyocyte cell states, and that we found, and I'm going to talk to you about those CM1 and CM2, just to keep it simple. In the control heart, most cardiomyocytes are in the CM1 cell state. There are a few CM2, mostly in the septum and also in the subendocardial region of the right ventricle, so not really in the pumping, particularly the pumping function of the places in the heart. In YAP5SA, CM1 is almost completely converted to the CM2 cell state. So that was a nice finding for us. So we went back, and then if you go in and look at the cell states, we know that the CM2 has basically two characteristics. It has very strong evidence for sarcomere disassembly, so that dedifferentiation thing that I mentioned. And just basically look in gene expression, you can see that there's a lot of evidence for different types of cytoskeletal genes, and we interpret this to be sarcomere disassembly. And then also we see downregulation of oxidative phosphorylation. So there's a very strong shift in the metabolic state, a metabolic reprogramming of those cardiomyocytes when you express YAP5SA in the cardiomyocytes. The other thing we found was that there was a very strong enrichment in the YAP5SA hearts for macrophages, which is what we're looking at right there. Is you can go in and look at the spots, and you can look very carefully at each spot and try to understand what are the cells which are surrounding your cell of interest. So now we're interested in CM2. So we went into the spots and looked carefully for what were the other cell types which were enriched with CM2. And we did this in the control and also in YAP5SA. And what we found was that there were cells that were expressing complement pathway genes, and they were very highly co-localized with CM2. So a little bit of a surprise. Complement is, innate immunity and inflammation and so on. But I'll take you through this. What we found is that there was an enrichment in YAP5SA for this gene C3. So this complement factor 3. Main component of complement cascade. And then I'll just focus on this gene called C3AR1, which is a receptor for C3. So if you look at down here at this right here, so we found that the cells that are actually expressing C3 were the fibroblasts. So fibroblasts are expressing C3, and this is strongly enriched in YAP5SA in a context of tissue renewal. If you will. So we're modeling low hippo activity and a lot of YAP activity. And then we found that C3AR1 and likewise C5AR1 were in macrophages, and that these were also more so in the YAP5SA hearts. And if you look at the spatial data and go back and do co-localization analysis, and you can be, very rigorous about this depending upon what your statistical cutoffs are, we found that the 3 cell types were highly co-localized in YAP5SA hearts, but not in the controls. And here's on this plot over here, you can see that that's quantified. So the way we think about this is that there are 3 cell types. There's the cardiomyocyte 2, there's the macrophages, which are expressing C3AR1, and then there's the fibroblast, which is expressing complement factor 3. So the cellular triad, which is characteristic of a microenvironment which is pro-regenerative and pro-renewal. So YAP5SA is very artificial. I love it, but not everybody loves it. So we went back and looked at some available data in neonatal mouse regeneration context, so the endogenous regenerative context of a mammal, of a mouse. And we basically took the spatial transcriptomics data from this lab, data from Cui et al. From the Olson lab, published in 2021, and we used our data to score their data. And what you find is, just to go through it quickly, is that in P1 hearts, so these are regenerative stage mouse hearts that had an MI, and you look 3 days later and 7 days later, we can see co-localization of the cellular triad right around where the infarct is. So those are the dark spots at that 3 days later. And by 7 days, when the infarct has started to heal, or almost completely healed actually, that those cell states have reverted back to baseline. We think that this is a YAP-driven event based on our YAP5SA cardiomyocytes in the adult, the experiment that I was telling you about. And so we scored the same data for YAP activity. So we know all the YAP target genes, and we can put YAP scores on all of the cells in the spatial data. And you can see that likewise, the triad, you can see that there's a lot of YAP activity, and it likewise, as it goes away, as the wound goes away, then there's less YAP activity. If we have the cellular triad, so we're claiming that these 3 cell types are somehow important, and they have to be co-localized and be talking to each other is sort of the intuitive model. And so we've done a lot of experiments now, but here's one of the experiments that we did early on. We expressed YAP5SA and in a mouse that had a C3AR1 deletion. So these are mice which are outside the regenerative period. YAP5SA in those cardiomyocytes, then typically, shown right here, you would get cardiomyocyte proliferation. And we quantified that looking at this marker of G2M CDK1. So there's quite a bit of proliferation when you express YAP5SA. when you do the same experiment in a C3AR1 mutant, so these mice don't have signaling through that C3AR1 receptor. There's a lot less evidence for proliferation. So CDK1 positive cells go way down in the mice that are mutant for C3AR1. I think what I think is also quite interesting, one of the things that must happen for a cardiomyocyte to proliferate and for cell renewal to happen is that they have to undergo sarcomere disassembly. And you could see in the controls, when you have a lot of YAP5SA in the cardiomyocytes, you have these cardiomyocytes which are undergoing sarcomere disassembly. We have a way to quantify that. We have a disassembly score. And basically, these cardiomyocytes that have a lot of dark areas at the central location, those are the disassembling cardiomyocytes. Right there's a nice one right there. We quantify it, but in the C3AR1 mutants, essentially sarcomere disassembly is really greatly reduced. So even in a context of a cardiomyocyte that has a lot of YAP5SA, so we're really strongly manipulating its cell state. If you don't have macrophages that are expressing C3AR1, you really stop the process. You don't get sarcomere disassembly, and you really inhibit progression through cell cycle. And so that really points to this fact that it is truly a microenvironment. There are multiple cells involved in this regenerative process. It's not only myocytes. We've done these other experiments to validate that further. Here's a loss of function study that we've done, looking at the C3AR1. We've turned back now to neonatal mice. So these are mice that would normally regenerate. If you infarct the mice at these early stages, I guess this was done at P4, so within the regenerative stage, and then you follow the mice out for more or less 4 weeks and see whether or not they can regenerate the heart. If you don't have C3AR1, so a C3AR1 mutant, you can see that the infarct doesn't heal. Even though normally it would, you could see that the controls do heal because, again, we did that manipulation in the regenerative stage. Ejection fraction. So right there, they don't recover their ejection fraction. Likewise, for C3, we did the same thing. So C3, this was a germline mutant that was at the time that is all that was available. So we did this experiment and we infarcted the mice. This was a P2. And then again, within the regenerative stage, basically the same experiment that I just told you about. And likewise, these mice also don't regenerate. If you do it in the regenerative window, the controls do regenerate. And then here you can see quantification. They don't recover their ejection fraction, whereas the controls do. And then you have more scar. So C3 is complicated in the sense that a lot of it is coming out of the liver, and it's in your serum and plasma. So we've made conditional mutants, and we're doing those experiments right now, specifically inactivated in fibroblasts, cardiac fibroblasts. But we didn't have that at the time, and so we did a few more experiments to look at this in a slightly different way. So we had those mutants, the C3 mutants, and we have infarcted them, and we did single cell to look at this question in a little more depth. And you could see that in the control, there's a group of fibroblasts which are expressing C3, and we have it marked there as C3 positive. But in the C3 mutants, the cell state still exists, but you can see that there's no more C3. And then if you go in and look at the difference between these two, focusing on those two different types of cardiac fibroblasts, that there are differences in that the C3 mutants have changes in their metabolic state. So they have more oxidative phosphorylation. Another way to think about that is that they have less glucose metabolism, so less glycolysis and things like that. We looked at that on a seahorse, a functional seahorse experiment. We isolated the fibroblasts, and you can see in the C3 mutants compared to the controls that there's much more ATP coming from mitochondria as compared to glycolytic ATP derives energy. And so this is consistent with this idea that, well, let me just say the fibroblasts, when they get injured, they have to change their metabolic state and become more glycolytic or use more glucose. So the way that we interpret this is that these C3 mutant fibroblasts don't make that transition. So they don't move as efficiently into the glucose metabolism. And so there's a couple of different possibilities. Maybe the fibroblasts don't sense the injury, or maybe C3 somehow within the fibroblast is important for that metabolic reprogramming. But it certainly is a sort of non-canonical function for C3 that we're looking at further, and it's consistent with some literature in the complement field, and we have talked to the complement field people, and there is emerging evidence for these non-canonical functions for complement. Okay, so here's our working model for this stage of the next of a pro-renewal heart. You have the CM2 cell state, and that has all those things I talked to you about. It has ability to proliferate, and it changes sarcomere, undergoes sarcomere disassembly, but also it makes factors. One of a direct YAP target is CSF1, and we know CSF1 can talk directly to those macrophages, and these C3AR1 macrophages that I told you about are actually tissue resident macrophages. We know that. We've done a lot of profiling of these. We also know that they make IGF-1, and we think that signaling back from the macrophages back to the myocytes helps change the metabolic state of those myocytes and makes it more glucose utilizing. The other important cell type is the C3 fibroblasts, and we would argue that the main function for C3 fibroblasts in this whole model is to provide activated C3 to ligate that C3 AR1 receptor and change the cell state of those macrophages. And so, we're looking at this now, in adult context and really delving more deeply into this using different types of conditional alleles now that we've acquired all the tools that we need. We touch upon some of the stuff that we are doing in congenital heart disease. So kids with different types of congenital heart disease. We published this paper in 2022 now, where we looked at many different types of congenital heart disease, structural as well as cardiomyopathies. And you probably can't read that, but it's not that important that you do. It's just a bunch of different, it was really a first shot across the bow for us to look at this problem. And we did single nuclear RNA-seq, and we looked at hundreds of thousands of nuclei, and we can identify all the different types of cell types in the heart, including rare cell types like mast cells and mast cells right there. We also and many inflammatory cells. So we definitely, we're very interested. It was very informative. Gave us a lot of insights into different types of congenital heart disease. But we wanted to look at this more carefully in a particular type of congenital heart disease called single ventricle. So we've been profiling hypoplastic left heart syndrome. So these are kids that are born with only one ventricle. Typically, what we've been profiling are kids that have a single, have underdeveloped left ventricle, so they only have a right ventricle. And these kids, because they only have one ventricle, that single ventricle is under an enormous amount of stress. And it provides us really a great opportunity, for us to try to understand what happens to a human ventricle when it's under enormous stress. In order to live, these children have to undergo staged surgeries the surgeons call it palliation because it's not curative. At the Norwood stage I'm going to be talking about it as stage 1. So this is immediately after birth. This is before the kids are in heart failure. So they were just born, but they're not in clinical heart failure. And then these other groups of patients, stage 2 and stage 3, they've failed at different points in the palliative surgery progression. There's something called the Glenn operation, and then the final Fontan operation to replumb all the great arteries and make this single ventricle more effective in perfusing the body. And so some of the kids fail at this stage 2, even before the Fontan, and some make it out a little bit past the Fontan. And then we also have some patients that receive the ventricular assist device. So the ventricular assist device is a pump that will unload the heart and improve hypoxia, as well as the volume overload that these kids' hearts typically see. And then we've done a lot of different studies. These are the different patients. We do have 3 paired pre and post VAD patients. That gives us a little more statistical power to compare the same patient at the time of VAD placement and at the time when they're getting a transplant after they've been on VAD for a various amount of time. Cardiomyocytes have very strong signatures of aging and senescence. Even at birth, they're born, that very first stage 1 with established senescence, and some of the differentially expressed genes are shown on this heat map, and some of these genes are involved in senescence and aging. This FOXO is a famous one. Here's how we have looked at this. We have an aging module, gene expression module for aging that we had devised in that 2022 Nature paper, and we applied that here, and you can see that the controls have a nice linear line between our scoring system and their actual age. But the kids with HLHS are older, essentially, at least in these early stages of their life. Here's some senescence scores. So you can take the cardiomyocytes in this case and put senescence scores on those cardiomyocytes. And even at stage 1, they have evidence of senescence. And then likewise in all the other later stages, 2, 3, and then even in post-VAD. And these are the different types of cardiomyocytes, typical sort of clustering that I'm not going to go into, but we see in the HLHS kids, we see enrichment for CM3 and CM2. So these are the sick cardiomyocytes that these kids have in their heart. Are quite distinct and, in our mind, scientifically anyway, from the scientific standpoint, quite interesting. Here's the UMAP, and we're very interested in this cluster called FB5. Just looking at differential genes between these different types of fibroblasts in the control versus hypoplastic left heart. We just jump over right here, the differential abundance. This FB5 is highly enriched in the kids who have the hypoplastic left heart. So there's a very, very drastic changes in cell state in the hypoplastic left heart, as you might imagine, considering the stress that these ventricles are under. So YAP is highly active in those FB5 cells in the HLHS cardiac fibroblasts. It turns out that YAP has one function in cardiomyocytes and has slightly different functions in the fibroblasts, and this is a question that we're wrestling with and trying to understand. But anyway, too much YAP activity in fibroblasts is typically a bad thing, and you can see that there's a lot of YAP activity in this FB5 fibroblast cell state. And one interesting thing that we discovered is these fibroblasts in the hypoplastic left heart kids are very similar to the fibroblasts that you would find in humans that have ischemic heart failure. Some of the data that's been published from the Kramen lab has identified those fibroblasts. So there is some similarity between ischemic fibroblasts and the fibroblasts that we find in these kids with hypoplastic left heart syndrome. We've looked at this question of YAP activity in those fibroblasts in greater depth. We can see that some of these are sort of the classic genes, YAP target genes, PTX3, and some of these other ones. And then this is a way to look at what are the transcription factor binding sites in those genes. It's called Regulon analysis, scenic in this case, and we can see that there's enrichment for TEAD elements in a lot of those genes. So that's consistent with this idea that the YAP-TEAD complex is driving activity. And then here's just showing you the TEAD. We see FB5 abundance is diminished after VAD placement. So you can see that FB5 abundance, so that bad FB5 cell state, high YAP activity, it gets very high, especially at stage 3. But then if you unload the hearts with the VAD, then you can see it really is quite reduced. And here, these are the pre and post VAD pairs, and you can see similar things. FB5 really the cell state is reversible, which is good, by unloading. So and so that gives us some thought about how we might think about developing treatments states. We use spatial transcriptomics. We know that the microenvironment really is where a lot of the important action is, and so we've done this in these kids on these tissues. We have some controls which were normal, but they're donors, and then we have stage 1, 2, 3, and then we have some post-VAD samples as well. What we're doing here, we're using the spots as our individual data points, and then we are looking for similarities between the spots to try to understand if there are, what's happening in the microenvironment, essentially. And that's, if you just get to the bottom panel right here, the different colors are the aggregates of the different clusters of spots, I guess is one way to think about it. And then if we do a UMAP for that, these are spot clusters, and I'm going to focus my attention on the C2 spot cell state or spot state. And if you look within those C2 spots, you can see that there's a co-localization of these different types of cells. Endothelial cells is the pericyte, fibroblasts, and myeloid macrophages are some of the most highly enriched. And these are all, I didn't show you the data, but all of those cell states are highly senescent. So not just the cardiomyocytes, but these other cells likewise are forming like these little cellular clusters, which are senescent. And you can look at this in a different way. This is maybe not that great of a way to look at it, but you can make a matrix. And if you focus on FB5, we've calculated the probability of having these different cell types being co-localized. So it's statistical calculation of the probability of those 3 cell types being co-localized in the spatial data. And if you look at FB5, you can see that FB5 is highly co-localized with EC4, PC2. That's a pericyte. This is a endothelial cell, which is also a senescent endothelial cell. And PC2, EC4, and FB5, right? So those different cell types are very strongly co-localized. And then next to lastly, if you just focus on the bottom, so these bottom 2, this is really the punchline. So if you take the spatial data and you look at co-localization of those cell types, and you do it with these density maps, you can see that in all these, this is the control, and all these kids that have sick hearts, that you can see co-localization of those cell types. And what we think we're seeing here is that there's a centrally localized co-localization of cell types, which has the strongest signature right there, those red signatures. And so the way what we think is happening is that there's a central group of cells, which I'm referring right here to as DNISH, but it's a centrally localized group of cells. And then if you move a few spots out, 3 spots in this case, from that central spot, that all of those spots likewise have signatures of senescence, although it's not quite as strong as the central spot. And then as you move further away into the remote areas, that those senescent signatures are gone. So if you want to, if we can quantify that and put it on a graph, these are senescent signatures. You can see that there's at the center, it's very high level of senescence. And as you move away from the center, this is the distance away. As you move further away from the center, those senescent signatures become less. In the post-VAD kids, post-VAD tissue, we see a similar sort of scenario, but it's less. There's a less amount, I guess, yeah, of those senescent signatures, but it's very similar. So VAD is good, but it obviously doesn't repair the tissue to back to normal, which is down here. So, in the senescence, there are these senescent cells make a lot of signaling factors, senescence-associated secretory proteins or phenotype, I guess some people call it. So anyway, they're inflammatory molecules. IL-6 is classic, and And so we think that that's the way. That's those center group of cells are making SAS factors, then signal out to the local microenvironment. And if you do the same thing that we did for the signature, senescence signature, and just look at the SASP genes, that you see a similar kind of thing. And in controls, you don't see that. So we think, we think that this analysis using spatial is really telling us about how these hearts fail. It's not necessarily that the whole heart or a region of the heart fails, but it seems like, at least in this context, that there are these little niches that start to fail. And presumably, as things would get worse, that those it would just continue to spread, and eventually, the whole myocardium would fail. But it looks like that there are these little niches, little spots of senescent cells which are causing the problem in hypoplastic left heart. So just to recoup what I've told you about cardiomyocyte regeneration, I think we have a good understanding now for how this is working. There's lots of data coming in from other labs in the world, my lab, but also other labs. And we know the sarcomere breakdown or disassembly is a critical event. Obviously, you need to make more cardiomyocytes. You don't want to make too many, obviously. And then inflammation, we know that you need this sort of pro-reparative tissue-resident macrophage population of cells is a good thing. Metabolic reprogramming, lipid utilization is bad for regeneration. There's a lot, our evidence, the evidence that I showed you, but now there's a number of papers from a group in Europe, Max Planck, Thomas Brown had published a paper showing that inactivation of CPT1B improves regenerative capacity of hearts. So that's a molecule that is important for lipid transport into the mitochondria. So there's metabolic reprogramming, and then there's some data now coming out about how redifferentiation might be. When you get dedifferentiation or sarcomere disassembly, there must be some mechanism to rebuild those sarcomeres, because obviously you need the sarcomeres for the heart to pump. Our HLHS data, we think that these children during development, not surprisingly, we know, during development, they're very stressed hearts. But now we're getting a handle about what exactly is happening. They're born with established senescence. And progression into heart failure involves these senescent niches that, maybe is a therapeutic target if we can manipulate some of these signaling factors that are extending these senescent niches, which we think are bad for heart function. And then the people who did the work, here are my collaborators. These are my computational collaborators, Xiao Li at Texas Heart, and Hassan Sami, who's at Baylor College of Medicine. And then Rich Lee did the spatial data, helped by Fanson and a number of these other people. Shiji now is independent in Cincinnati. Zhang and Bing did some work as well as Jeff. So I'll stop there. Yeah. Oh, yeah. There you go. Those koalas are very cute. But
thank you for the enlightening talk. It's really great that the finding from the development biology all the way to the heart treatment. So the podium is open, the stage is open for the question and answer. I highly encourage training to ask a question. I was a shy student, but please don't be so yeah.
Wow, that was outstanding. And I'm really glad I use your papers in my classes.
Thank you.
It's certainly transformed the way we think about cardiac regeneration and what we need to do to get it to happen. So in your YAP5 assay hearts,
yes,
have you looked at what happens to the other TEAD partner, VGL4? Because you mentioned at some point you want not just sarcomere disassembly and cell proliferation through 1 or 2 rounds, but you actually want the TEAD factors to go back to where they were, which is controlling cardiac gene expression, and, maintaining the cardiac differentiated state. So
Yeah.
so TEADs don't just bind to proliferative genes with YAP, but their other daytime job is to work with VGL factors to maintain the differentiated muscle state, VGL2 in skeletal muscle, and maybe even in early differentiating myocardium, but also VGL4, which is a repressor and keeps things quiet. So
yeah. Well, in YAP5SA, it's a great question. And we have, on our transcriptomics, we know that those vestigial VGL are upregulated. So they're very strongly upregulated. And, some of them are, I can't remember which one, but they're direct YAP targets. So it's part of this negative feedback loop, which is a very strong thing in the cardiomyocyte. The cardiomyocyte, the way we think about it, the way I like to think about it, the cardiomyocyte hates to have YAP in the nucleus because some of the top targets are, LATS2, vestigials, angiomotin. Angiomotin-like 2 is always way up. And so these are all negative regulators. Despite that, we still see a ton of YAP target genes being turned on. Even those vestigials are still there in the nucleus. And we're trying to understand that now. So there's a lot of cardiomyocytes in those hearts that don't have a lot of YAP activity. And so I am guessing that some of these mechanisms that you're referring to, may be helping to shut YAP activity down. So I think that's sort of the next thing that we're going to try to understand is what happens to those other cardiomyocytes that don't have a lot of YAP activity. It may be the negative feedback loop. There were some other interesting things that we're uncovering, which are a little bit unexpected, which, is my dream always to find unexpected things. But I think that's a great question, yeah.
I also noticed you left out an arrow, how the cardiac myocytes are talking to those C3 fibroblasts, because clearly if you're expressing YAP5SA in cardiomyocytes and the fibroblasts are doing something, they're not doing it, due to YAP expression. They're doing it in response to the cardiomyocytes and the reprogramming of the cardiomyocytes. So do you have
What's
anything?
Happening?
Yes, what happened?
Yeah, honestly, we area that we're still a bit unclear about what's happening we know that they're important. We know they're there. And we have the functional evidence that they're important. And I could say that we've done the conditional mutants now, and we're seeing phenotypes. So that's good, using fibroblasts. How all that signaling is happening with between the fibroblasts and the myocytes, and likewise macrophages, it's still something that we're working on. Yeah.
Direct, indirect?
I mean, a lot of it is direct. Yeah, sure.
So definitely. Gap junctions between myocytes and fibroblasts?
No, we don't see.
No,
we haven't seen
nothing like that.
We haven't seen anything quite as interesting as that.
Oh, okay.
If you think that's interesting, but
okay, thanks.
Those are good questions. Yeah.
Overturns the common belief that myocardium does not regenerate. Hopefully, in a few years, this will come to clinical use. Now, in your cell differentiation study of the cardiac cells, did you include those of the signal generating type, let's say in the synovial nodes?
We have not done that. And one of my former trainees, Dr. June Wong, who has taken that particular project to her own lab to try to look at what Hippo is doing in sinoatrial node. She had a circulation paper a few years ago, 2 years ago, I guess. And then also this idea that you can regenerate the conduction system directly. So we're not doing those studies because Chinese take their project with them, and so she's working on that. But I'm tempted to start working on it again because it's such an interesting question. But we haven't done it. But thank you for the question.
regeneration, but by making the cardiomyocytes regenerate and grow and differentiate, but how do you stop it? Because it has to have like something to say like, oh, okay, up to here. How do you control that?
Yeah, no, I mean, that's a great question. You know, in the in YAP5SA, which we've lost all that regulation or most of it, you know, they don't stop. And so those mice will eventually, well, those YAP5SA mice don't survive. So the YAP5SA is, like I said, is the sledgehammer, and that you can't really use that clinically. However, Salvador is our target that we have the company has licensed that. Salvador is a very, a relatively minor knockdown of the pathway. And so as the wound heals, YAP will be in the nucleus. YAP is in the nucleus when you knock down Salvador. And then as the wound heals, it turns on, the hippo pathway gets turned back on again because of the negative feedback pathway that I didn't show you a slide, but YAP turns on all these other proteins that turn YAP activity off. So as the wound heals and the organ size control pathway kicks in, then the cardiomyocytes stop proliferating. And I took that slide out. I should have left it in because we showed in the pigs that if you look at 30 days after the infarct, there's good evidence that the cardiomyocytes are still proliferating. But if you look at that 3-month time point later, there's no more evidence of cardiomyocyte proliferation. So that's because the wound has healed to a large extent and the stop signal has kicked back in. It's very important. There's a couple of things. So that's safety, right? But also it's important that we did RNA interference because RNA interference is a knockdown of the pathway. So that allows the cell state, the cardiomyocyte, to reverse itself. It's not a knockout. So the cardiomyocyte cell state can reverse itself as the microenvironment heals again. You know, I think that's something that we probably need to do more work on. I personally am very comfortable with it, but this is a question that investors always ask us, and I just about every time I give a talk, I get that. So it's an intuitive question, but you know, we're pretty sure that the negative feedback loop kicks in and turns the hippo pathway back up, right? Turns YAP activity off.
Did you notice or did you study the delivery site of, it's a virus, right? So
yes,
when you deliver it in the atria or in the ventriculum, do you notice the differentiation of the cardiomyocytes depending on where you deliver it?
Are you asking the question of whether there's regional differences in proliferation?
Yes.
In the heart?
Yes.
We didn't notice it in our studies. In the YAP5, not in the Salvador knockdown, we didn't notice it, but there's other evidence that there are regional differences, yeah. But we didn't really see it in our studies.
Okay. Thank you so much.
Thank you. Those are great questions. Thank you. Yes.
Yeah, if I can ask a question as well. This is a fantastic really tour de force, and you know, really look forward to the clinical studies, right? You know, in terms of translation. A couple comments and then a question. One is that I think it really underscores the I guess the sociology of the myocardium. You know, in that there are these, you know, kind of regional organizations in a way, you know, in which we don't tend to think about, right? You know, we tend to think of the heart as actually uniform.
Yeah.
But in fact, but it's probably these regional differences that actually allows, you know, some of the stress responses, you know, to be successful. And I think even, you know, a puzzle that we sometimes have is that, you know, how could a failing heart recover, for example, you know, using medications and things like that. And it's probably because not the whole heart actually has failed, right? You know, there are these regions that can recover, and that's actually driving some of those forces, you know. And now one area which we've been working a lot on is the senescence of the vasculature. You know, it's more in the context of a heart failure and also similar models. And just as a long time ago, we did actually, you know, work in congenital heart disease. In fact, you know, the senescence markers, this is more on kind of the cyanotic heart disease patients. Yeah, it's actually sky high, so very, very much parallel, through your observation. And of course, these patients, develop cardiovascular complications way earlier as well. So the couple questions I have are, one is, what is the kind of the sweet spot in terms of the amount of myocyte that you reprogram, right? Because too much, obviously, you get into problems, and you want enough to actually to have the impact, right? Because you're working on the border zone, but ultimately you do actually want to have the remodeling that's adequate, to
Right.
Yeah, and so what was from your experience, what is the kind of the sweet spot?
Yeah, I think that's a good question. I can say, minded people always ask me that. I can say a few things about that. So we know that in these YAP5SA hearts, which is a very powerful model, we see about 40% of the cardiomyocytes are dividing, based on these newer methods that have been published and how you look at, and we know that's too much. So that's definitely too much. In the Salvador knockdown, it's probably closer to, maybe at most 5%. At any one time. I will say one more thing, though. It's not just division, but it's also sarcomere disassembly.
Yes.
So one of the things that you'll notice in the YAP5SA, maybe you didn't notice, I didn't show it that well, but there's a lot of sarcomere disassembly. And so that also is very debilitating to the heart pumping function. It's not just dividing, but this disassembly step. And we've been doing studies, and we know that's the case because we've been doing studies. We have a reversible, I couldn't get the slides together in time for this, but we have a method now to turn YAP on and off, and it's very quick. It's using one of these splicing mechanisms. It's not tetracycline. It's a splicing mechanism anyway. It comes out of CHOP. Anyway, it allows us to really turn YAP on and off in about 72 hours. YAP5SA will stay on for about 72 hours. If we give a very high dose that induces the splicing and then YAP comes on, it looks very much like the data I was showing you. The mice die quickly, but we can titrate it now. And we know that at some doses, the mice will get sick and the hearts have a lot of sarcomere disassembly, but not that much proliferation, and they're still very sick. So I think that the thing that really kills these mice, obviously proliferation is not good, but it's probably that sarcomere disassembly. Yeah, they're just completely, they're just the cardiomyocytes just don't have that pumping function. So that's something that we're going to have to, I think we'll have to try to work with a little bit more. I think actually it's a target, a way to manipulate things also. You can, maybe if we can figure out ways to make sarcomeres disassemble, it might be useful. So anyway, that's a long-winded answer to your very important question that we're thinking about all the time. How many cardiomyocytes do we actually need? And so not that many really, because it's a microenvironment. It's about 5% probably.
Can't you put patients on a BiVAD while they're being treated with the YAP, the 5SA transient or the Salvador, and then while their sarcomeres are disassembling and the cells are repairing the damage, and then you're taking the load off the heart so that the afterload at least, and so you can let your patients recover. Because I mean, clearly, if you're disassembling sarcomeres in a mouse, the heart isn't going to be doing very well.
Right.
And
I mean, that's kind of the dream. But this, as clinical practice is a tricky thing to try to change. So, as an example, in congenital heart disease, those kids, the standard of care is they all get a transplant.
Right.
That's it. They don't look for people to get better on a VAD. Kids, adults is different, but in the pediatric population, once you get a VAD, pretty much you're going to get a transplant. So that's standard of care. And then you run up against similar things in the adult. But I think you're right. I think conceptually those are good thoughts. Yeah.
Wasn't there the case of a young woman who had an ectopic heart transplant and whose primary heart was left in place after myocarditis, and somehow there was recovery of the unloaded heart. I seem to remember that somewhere in the literature a long time ago.
Well,
Maybe.
myocarditis would be the context where that would happen.
Yeah.
Right. I don't remember that particular case, but yeah, I think if you can support people that have had myocarditis, they can recover function for sure.
My
No,
question is,
yeah. Okay, go ahead.
yeah, I think you can just, yeah.
So the YAP5 or earlier studies that those adult cardiomyocytes will revert back to a fetal, approximately E14.5 is what we published back then. If you just compared signatures of gene expression and get a cell state, so it's about E14, similar to an E14.5 embryonic mouse embryonic heart. So a little bit more later than mid gestation. And then what happens to nucleation? Well, you get enrichment for mononucleated cardiomyocytes for sure when you express YAP5SA. Those binucleated cardiomyocytes actually undergo that last step in cytokinesis. That's the most likely thing. Yeah. But that's not that easy to show, but that's still our hypothesis. But we're trying to figure some of those things out. That's more like live imaging type approaches, which are cool.
Okay, if there's any other question?
Okay.
Thank you very much again. And then please join me. Thanks, Dr. Martin. Yes. So now we are going to have a health break until 10 more minutes, I think. Yeah, 2:30, 2:35, 2:30. Okay. All right.
Really quick bathroom break, so we can get you back on track. Okay. 1 presenter online.
I know.
Is it going to be okay?
Yeah, it's gonna be okay.